Chapter 7 covers the different types of cellular respiration. In this first screencast, I'll introduce the different types of cellular respiration, and we'll look in detail at aerobic respiration. All living things must do some type of cellular respiration. It's how energy from organic molecules is made available to organisms to use. The term cellular respiration refers to the chemical reactions used to release energy stored in organic molecules. Cellular respiration and breathing are two different processes. In organisms that use aerobic respiration, the two processes are linked. Breathing, whether it be moving air in and out of lungs or moving water across gills, involves exchanging oxygen and carbon dioxide between the organism's tissues and the surrounding environment. But not all organisms use aerobic respiration. Anaerobic organisms don't have to breathe. There are two main categories of cellular respiration: aerobic cellular respiration, often short to just aerobic respiration, and then the second type is anaerobic respiration, which includes two different types of fermentation. Most of the organisms that you're familiar with use aerobic respiration. It's called aerobic because it requires oxygen gas. In terms of the amount of usable energy captured per molecule of glucose used, this is by far the most efficient form of cellular respiration. All large and/or active organisms use this. They may switch to other pathways for short periods of time, but they quickly must return to using aerobic respiration to meet the energy requirements of their bodies. Anaerobic respiration does not require oxygen. Some anaerobic organisms cannot survive in the presence of oxygen gas. Most aren't harmed by oxygen, they just don't need it. Your body can switch to a type of anaerobic respiration for a short time. It's called lactic acid fermentation. Let's look at some details of aerobic respiration. We're going to use glucose as our energy source. As far as I know, all organisms can use glucose for energy. When using glucose, the first series of reactions is called glycolysis. The name means sugar splitting. Glycolysis basically splits the glucose molecule in half. Chemical bonds are broken and the glucose is oxidized, releasing energy and electrons. The carbon atoms in glucose end up in two molecules of pyruvate, a 3-carbon molecule. Looking at a little more detail in the first part of glycolysis, glucose is a relatively stable molecule. To get it into the mood to react, we have to first invest some energy. 2 molecules of ATP supply the energy to split glucose into 2 smaller, more reactive molecules called G3P. The 6C reminds you that glucose has 6 carbon atoms. The 2 G3 molecules each have 3 carbon atoms. In my diagrams, the red arrows pointing from ATP to ADP indicates that we have used some energy. In the second half of glycolysis, the G3P is now converted to pyruvate. This involves oxidation, in which the G3P loses electrons and energy. The electrons and some of the energy they carry is used to reduce NAD, making NADH. NADH plays a similar role to ADP-ATP. It transfers energy from one place to another in your cells. Rather than breaking or making bonds between phosphate groups, as in ATP, NADH carries high energy electrons. In addition to creating 2 molecules of NADH, the second half of glycolysis also releases some additional energy that's captured in the form of ATP. My green arrows indicate that usable energy is being released. This slide summarizes glycolysis. 1 molecule of glucose is split, producing 2 molecules of pyruvate. The usable energy that was released has been captured in the molecules NADH and ATP. There's still a lot of energy available in what's left of the glucose, in other words, the 2 molecules of pyruvate. Pyruvate is next used in the preparatory reaction, sometimes also called the transition reaction. Each pyruvate is oxidized as a carbon is split off. The electrons and energy released are captured as NAD is reduced, forming NADH. 2 of the carbon atoms from the pyruvate are now in the acyl group that attaches to coenzyme A. And the 3rd carbon, which was split off from the pyruvate, is released as carbon dioxide. The acyl group produced during the preparatory reaction next enters into the Krebs cycle, also called the citric acid cycle. This cycle produces a lot more NADH. Another nucleotide similar to NADH, called FADH2, is also produced, and a little bit of ATP. This is also when most of the carbon dioxide produced during aerobic respiration is released, twice as much carbon dioxide as during the preparatory reaction. It's called a cycle because OAA initially reacts with the acetyl group, and at the end of the cycle, OAA is regenerated. This is my attempt to summarize the citric acid cycle. The two acetyl-CoA molecules from the preparatory reaction enter into the cycle. Coenzyme A releases the acetyl group, and this acetyl group reacts with OAA to form citrate, also called citric acid. The citrate is then oxidized, and two carbon atoms are split off from each citrate. Some of the energy released is captured in the conversion of ADP to ATP. Much more of the energy and electrons are captured as NAD is reduced, forming NADH. In a similar fashion, FAD is reduced, forming FADH2. The carbon atoms split off from the citrate are released as carbon dioxide, and then OAA is regenerated, and the cycle repeats. The majority of the energy released so far in aerobic respiration is now in NADH. Your cells need that energy in the form of ATP. The last series of reactions in aerobic respiration is called the electron transport chain. It uses the NADH to make a lot more ATP. The oxygen gas used during aerobic respiration is used in the very last step of the electron transport chain. I think of the electron transport chain as kind of like a game of hot potato, with electrons being the hot potatoes. NADH is oxidized as it's converted back to NAD. The electrons are passed through a series of molecules which are alternately reduced as they accept the electrons, and then oxidized as they pass the electrons on to the next receptor. Oxygen gas serves as the final electron acceptor. It combines with electrons and hydrogen ions to form water. As the electrons are passed from molecule to molecule, energy is released in a stepwise manner. This energy is used to make 3 ATP molecules. FADH2 is used in a similar fashion. It gives its electrons to a later molecule in the chain, and as a result only produces 2 ATPs. Let's look closer at how the ATP is synthesized. Cells use a process called chemiosmosis. Unlike osmosis, where water moves across a membrane, chemiosmosis involves the movement of hydrogen ions across the inner membrane of the mitochondria. An enzyme, ATP synthase, is able to use the movement of the hydrogen ions as a source of power to generate ATP. This diagram represents the inner membrane inside the mitochondria. The blue structures are the molecules which make up the electron transport chain. As electrons are passed from NADH or FADH2 and on along through the electron transport chain, hydrogen ions are pumped out into the inner membrane space. Think of these hydrogen ions as kind of like water backing up behind a hydroelectric dam. The enzyme ATP synthase functions sort of like the turbines in the dam. As hydrogen ions come rushing in through the ATP synthase, a third phosphate group is added to ADP, making ATP. This slide summarizes aerobic respiration. The 4 sets of reactions are glycolysis, the preparatory reaction, citric acid cycle, and the electron transport chain. Glycolysis uses glucose and produces 2 pyruvates, 2 NADHs, and a net gain of 2 ATPs is realized. In the preparatory reaction, pyruvate is used and produces 2 acetyl-CoAs, 2 molecules of carbon dioxide, and 2 NADHs. The citric acid cycle uses the 2 acetyl-CoAs and produces 4 molecules of carbon dioxide, 2 ATPs, 6 NADHs, and 2 FADH2s. Finally, the electron transport chain uses the NADHs and the FADH2s produced in the previous reactions to make a lot more ATP. Doing some bookkeeping, 2 ATPs are formed directly during glycolysis and the citric acid cycle. A total of 10 NADHs are produced for each of these NADHs, which enters into the electron transport chain. 3 ATPs are produced. The two FADH2s from the citric acid cycle also give their electrons to the electron transport chain and produce a total of 4 ATPs. Adding all those together gives a total of 38 ATPs. You might remember in lab, I told you aerobic respiration produces 36 ATPs. What's up with that? This diagram is from your text. It's similar to my summary on the previous page, except that it emphasizes the fact that glycolysis occurs out in the cytoplasm, while the rest of aerobic respiration occurs in the mitochondria. In most of your cells, active transport is required to move the NADHs produced during glycolysis into the mitochondria, where they're used in the electron transport chain. That active transport uses 2 ATPs. So, 38 minus 2 ATPs for the active transport gives a net gain of 36 molecules of ATP per glucose. Has all this chemistry fried your brain? Let's take a break. In the second screencast for Chapter 7, we'll tackle anaerobic respiration.